Yes, students, yesterday I have completed the chapter Microbes in Human Health, and that was belonging to unit number 3, wherein you studied only 2 chapters. Unit number 3, Human Health and Disease, chapter number 8, and chapter number 10, Microbes in Human Health. Chapter number 9 is a deleted portion. It is not for your examination. Thereafter, you are moving to unit number 4, and that is Biotechnology. And we reach chapter 11. Chapter number 11, and that is Biotechnology Principle and Process. Biotechnology Principles and Process. Biotechnology Principles and Process. First of all, how to understand what is biotechnology. As you know, the word bio means life. Now, and technology means it's a technique of various fields of science like physics, chemistry, or any type of technical of engineering, even the non-engineering technique. All that, if it is used and implemented in our biological science, and that is what you call as a biotechnology. Nowadays, you know, IT and BT you are listening always, information technology and biotechnology. How information technology is widespread, in a same way, biotechnology is also nowadays is become more and more advanced, and only by biotechnology it is possible to get a quality quantitative biological product, and what we can get our own desired product as well. ನಮಗೆ ಯಾವುದು ಬೇಕು, desired ಅಂದರೆ ಯಾವುದು ಬೇಕೋ ಆ ಪ್ರಾಡಕ್ಟ್ ಮಾತ್ರ ತಗೊಳ್ಳಿಕ್ಕಾಗುತ್ತೆ. That is nowadays advancement in what here biology, and that is why here in biology we are nowadays using so many different type of techniques of various field, and by that it is possible to produce desired things, desired product, desired component, desired chemicals, desired the enzymes, or it may be a protein, or it may be a type of hormone. Nowadays, you know, in the market antibiotics are there, antibodies available, hormones like insulin is also available. No, it is used to treat the diabetic patient, and some enzymes are also available. No, many drugs, different type of drugs, alkaloid, etc., that all synthetic, etc., all can be nowadays get by biotechnology. Then in industries also, pharmaceutical industry, then in some chemical manufacturing industry, wherein also our biotechnological process used, and that to be in the preparation of vaccine and many more field where biotechnological process is used. There are several advantages of this biotechnology, and this biotechnology not only in production of chemical enzymes, etc., and nowadays biotechnology also variously used in agricultural science also. This is what the agriculture also. Agriculture kuda biotechnology here used variously, and that to be in the production of rice, jawar, then wheat, and then vegetable, then fruits, so many things, and that to be get quality and qualitative, one. And then the second thing is, and as much as possible to reduce the usage of the chemical. As you know that nowadays we are using more chemicals in the field for agriculture farming, like insecticide, fertilizer, pesticide, herbicide, so many that. And without using how best we can create a new species of plant by biotechnological process. Seedless fruits are there. Correct. Seedless fruits also you know. Now, best quality protein and carbohydrate containing biofortified food is also available. Biofortified ಅಂದ್ರೆ quality containing protein, then lipid and then vitamin rich, vitamin A, vitamin B, vitamin C rich and then iodine rich ಅಂತ ಫುಡ್ ಅನ್ನು ಇವತ್ತು ನಾವು ತಯಾರು ಮಾಡ್ತಾ ಇದ್ದೇವೆ, which is not available to the poor person also. They cannot get the protein rich food easily in the market. For example, egg is costly. They can't eat every day egg. No. So iodine containing food they can't get very easily like fruits. No. So if all it is included in our daily diet ಅಂದ್ರೆ rice, particular rice ಮತ್ತು vegetable, ಈ ರೈಸ್ ಮತ್ತು ವೆಜಿಟೇಬಲ್ದಲ್ಲಿ ಪ್ರೋಟೀನ್ ಜಾಸ್ತಿ ಇದ್ದರೆ enzymes if it is there, if enzymes also if it is there, and iodine is also available. Iodized the rice and many the vegetable if it is prepared, they need not require to go for other fruits, other vegetable, costly fruits and costly vegetable. In the rice only these are available, and they can get what is that scarcity is there. No, what that scarcity is that they can get from their own daily food only. So that is why here what in the agriculture also it is what variously used and very common biotechnology, the process that is used, very common biotechnology process that is used is the genetic engineering. I think you are very familiar about this. Genetic engineering. What is the meaning of the engineering, you know? Engineering means it is one type of technology wherein some what the process used and then to cut, then splice and then rejoint, isn't it? ಇದಕ್ಕೆಲ್ಲ ಒಂದು ಟೆಕ್ನಿಕ್ ಬರುತ್ತೆ. Just like repairing the cycle, repairing the bike, repairing the car, how the automobile engineering you must know. Similarly, electrical engineering, electricity supply, etc. No. So like the civil engineering, how to build the house and all that, and what is required. Exactly same here with the genetic engineering is also what important raw material require, components require, but remember that takes place at the level of gene. That all taken place at the level of the gene. Yes, now you are very familiar about the word gene. What is a gene? Gene is the segment of the DNA. Gene is the segment of the DNA. You see here. This is a DNA and here a particular segment of the DNA. And see this much is and that is considered as a gene. And you know that in a segment of DNA on the chromosome, there is a two main chromatin bodies there. That is one is a euchromatin, another one is a heterochromatin. Euchromatin means what? So euchromatin means a genetically active part, and it will go for protein synthesis. And heterochromatin means what? Genetically inactive part that may not carry the protein synthesis. Again, this portion, a segment of DNA, this is called as a gene. And this gene, if it is taken for any technique, related technique, and that is what you call as a genetic engineering. ಅದಕ್ಕೆ ನಾವು ಏನಂತ ಹೇಳಿದ್ರೆ genetic engineering. And the genetic engineering in which here different type of technique is there, and that the technique like here the recombinant DNA. All you studied in online class recombinant DNA technology. It is rDNA, or maybe it is a gene cloning or gene transferring. Okay, so all these are the different type of technique that is used in what here genetic engineering. and that depending upon the what our aim. What is our aim? What we are going to do? What we are going to create? What you are going to produce? No, all that depend. If our intention is to produce a protein or to generate protein, or we require insulin or require enzyme, and then which host we have to use, which is a desired part of the gene. Now, how then they have to cut and splice. All these are the some technique that all you are studying in what? The genetic engineering and the gene level. Then the question mark is it that much of easy that what we are saying, just like automobile engineering, civil engineering, then mechanical, so many that engineering is there, wherein all that parts, the raw material, then tools we can see in the naked eye. No, ನಮ್ಮ naked eye ದಲ್ಲಿ ನೋಡ್ಕೊಂಡೆ ಮಾಡೋದು. But is it like that? No, this is not like that, because gene is a microscopic, super microscopic. You can't see them in the naked eye. DNA only you cannot see. DNA ನಿಮಗೆ ಕಾಣೋದಿಲ್ಲ. Then how you can see the gene? So means these all are taken at the micro level. You cannot imagine in our body. These all what process is going without our knowledge, and that you cannot compare with today's any what advanced machinery, advanced machinery or any device. What I am telling you, understanding what that process is going in our body. Now all physiological activities what is going in our body, that you cannot the compare to any devices of the world. That type of efficiency, okay, then fast accuracy that is maintained in our body with what 0.0000 what error. If any error in this process, and we may go to the abnormal condition. And you just imagine, ನಮ್ಮ ಬಾಡಿಯಲ್ಲೂ ಕೂಡ self genetic engineering is also one type. Understood? So means here you studied the DNA replication, you studied, isn't it? Then DNA as a genetic material also you studied. Then RNA world also you studied. Correct? And wherein all you studied, how fast the replication taken place, in what way it has taken place, that all you studied. So just you cannot imagine ನಮ್ಮ ಬಾಡಿಯಲ್ಲಿ what and what process taken place, what and what things is going every day, and that should be very smooth, and that to be into 24/7. Early you stop, there is no stop. If it is stop, and our life stop. You cannot imagine our ability of the brain. No, and whenever you buy the mobile or computer, then you ask storage capacity. 8 GB, 10 GB, 12 GB, 16 GB, like this, on that base storage capacity. Is there any such storage capacity to our brain? How much things it retain in our brain you can? Yes, storage capacity is full, and yes, I can't recall and memorize the old one and the no. You cannot imagine our brain capacity. 1400 odd cc our brain capacity. Any amount, any quality, any quantity, but it can take, and as and when it require, it recall also. That type of engineering process also taken place in our body. But what I am telling here, how the genetic engineering can be undertaken and what is the advantage, principles and the process that you are studying here. And now that we continue here in the principles. As you know, see here, when the segment of the DNA acts as a gene, and during the the activation of the gene, and every gene having, or every DNA having, there is one small point is there. Point again, there is the origin. And then the point is called the origin of replication. That already we studied. origin of replication. No, and that origin of replication is very important. ಅದು ಇದ್ರೆ ಮಾತ್ರ ಜೆನೆಟಿಕ್ ಇಂಜಿನಿಯರಿಂಗ್ ತೊಗೊಳ್ಳಿಕ್ಕೆ ಆಗ್ತದೆ. ಅಂದರೆ the used what the desired piece of DNA has an ability to what the replication ಆಗಲಿಕ್ಕೆ ಅದೊಂದು ರೆಪ್ಲಿಕೇಶನ್ ಆಗುವಂಥ ಒಂದು ಕ್ಯಾರೆಕ್ಟರ್ ಅದಕ್ಕೆ ಇರಬೇಕಾಗ್ತದೆ. ಆ್ಯಂಡ್ ದಟ್ ಇಸ್ ದ ಒರಿಜಿನ್ ಆಫ್ ರೆಪ್ಲಿಕೇಶನ್. The property is very very important, and that replication fastens the process and it gives qualitative and quantitative product. So that is why here nowadays biotechnology and the genetic engineering process become very popular in all the fields. Ella field allu in the genetic engineering is nowadays used, and that is why for biotechnology or genetic engineering or gene transferring for anything we require some important tools. See, tools of genetic engineering. Tools of genetic engineering. Here the tools of genetic engineering andre. So tools means the requirement. ಒಂದು ಎಂಜಿನಿಯರಿಂಗನ್ನು ಒಂದು ರಿಪೇರಿ ಮಾಡಬೇಕು. ಆಟೋಮೊಬೈಲ್ ಇಸ್ ದರ್ ಅಥವಾ ಕಾರ್ ಇಸ್ ದರ್, ಯು ಹ್ಯಾವ್ ಟು ರಿಪೇರ್. ಬೈಕ್ ಇಸ್ ದರ್, ಯು ಹ್ಯಾವ್ ಟು ರಿಪೇರ್. ಆ್ಯಂಡ್ ದೆನ್ ಸೈಕಲ್ ಆಲ್ಸೋ ಯು ಹ್ಯಾವ್ ಟು ರಿಪೇರ್. ಆ್ಯಂಡ್ ಫಾರ್ ದಟ್ ವಾಟ್ ಯು ವಾಂಟ್ ಸ್ಪೇರ್ ಟೂಲ್ಸ್ ಬೇಕು ಅಲ್ವಾ? ಟೂಲ್ಸ್ ಲೈಕ್ ಸ್ಕ್ರೂ ಹೋಲ್ಡರ್, ದೆನ್ ಸ್ಪ್ಯಾನರ್ ಆ್ಯಂಡ್ ದೆನ್ ಹೋಲ್ಡರ್. ಸೊ ಮೆನಿ ಥಿಂಗ್ ಈಸ್ ರಿಕ್ವೈರ್ಡ್. ವಿದೌಟ್ ದಿಸ್ ಯು ಕಾಂಟ್. ಕೈಯಲ್ಲೇ ತಿರ್ಸ್ಲಿಕ್ಕೆ ಆಗೋದಿಲ್ಲ. ಎವ್ರಿಥಿಂಗ್ ಯು ಕೆನಾಟ್ ಡೂ ಬೈ ಹ್ಯಾಂಡ್. And for that requires some tools. And when that all the proper tools are there, then only you can open, then repair and then refix. Tools illada you become handy. Now similarly in genetic engineering, gene what here engineering, what is that you studied just now, the gene transfer, gene cloning, RDNA technology, all the same, all genetic engineering. And that require here tools, and the first tool is enzyme. First tool is an enzyme, and then second is a host. Second is what? Second, what the next one is the plasmid. But better you write host only. Host, and then third one is a plasmid, or it is a vector, what you call as. And the last one is bioreactor. See, it may be asked in the 2 marks question. Mention the tools of genetic engineering. Only the 4 names you have to mention: enzyme, the host, then the plasmid as a vector, and then last one is what? The bioreactor. Okay. See, enzyme, as you know that enzymes are commonly called as a biocatalyst. They foster the rate of biochemical reaction. You studied in the previous year enzyme topic, and their importance is more in all the biological process. ಯಾಕಂದರೆ ಅದು ರಿಯಾಕ್ಷನನ್ನು ಬೇಗ ಮಾಡಿಕೊಡ್ತದೆ, ಫಾಸ್ಟ್ ಮಾಡಿಕೊಡ್ತದೆ. ಈಗ 1 ತಾಸಲ್ಲಿ ಆಗುವಂಥ ರಿಯಾಕ್ಷನನ್ನು ಬರೀ 1 ಸೆಕೆಂಡ್ದಲ್ಲಿ ಮಾಡ್ಬೋದು. Because of this reason only nowadays we are carrying in time all physiological, ಅಂದರೆ our body carrying in time all physiological process, and we are normal, and so that we can carry our everyday's activities. ಇಲ್ಲ ಅಂದರೆ we have to be on in slept condition always. ಯಾವಾಗಲೂ ಮಲಗೇ ಇರಬೇಕಾಗ್ತದೆ. We can't do anything, and that what you can see in old age, what the person, old age personಗಳು ಹೇಗೆ they can't move more, they can't walk after the age of 70, 75, 80, that depending upon the person, not all, depending upon the person. So their body slowly slow down the all physiological process because of the inactivity of the cell. Okay, ಇಲ್ಲಿ what the enzyme are the most important. How best enzyme working in our body and cells also active. Similarly, how best enzyme work in genetic engineering, and result we will get the better. So there are 2 type of enzymes are there here, and that is not 2 type. The enzyme is generally called as restriction enzyme. Enzyme and called restriction enzyme. See, the name itself indicates here restriction. There is some restriction is there, specificity is there. No, and that enzyme cannot operate anywhere and elsewhere type ಹೀಗೆಲ್ಲ, and there is a restriction is there, and that restriction enzyme is a two type. There is a first one is restriction endonuclease, and another one is restriction exonuclease. Both are REN. ಎರಡಕ್ಕೂ REN ಅಂತಲೇ ಕರೀತಾರೆ. ರಿಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸ್ and restriction exonuclease. However, what is this difference? First of all, restriction ಅಂದ್ರೆ restriction means always this enzyme operate on the restricted sequence of the DNA. You are studying genetic engineering, so genetic engineering means it is what here DNA ಅಂದ್ರೆ DNA ಮೇಲೆ ಮಾಡುವಂತ engineering. Segment of the DNA is called as what? The gene. So there is this much portion if you have taken, ಈ portion ನಾವು ತಗೊಂಡಿದ್ದೇವೆ and here for GAATTC ಇದ್ರೆ now and here is also GAATTC. This is 5'3' and this is 5'2'. This is the sequence. In this sequence, a particular restriction enzyme operate. ಅಂದರೆ ಈ ಸೀಕ್ವೆನ್ಸ್ಗೆ ಒಂದು ರಿಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಜೈಮು, ಇನ್ನೊಂದು ಯಾವುದೋ ಸೀಕ್ವೆನ್ಸ್ ಇದೆ ಅದಕ್ಕೆ ಒಂದು ಬೇರೆ ರಿಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಜೈಮು. ಅಂದರೆ ಆಲ್ ರಿಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಜೈಮ್ not all restriction endonuclease, they may not operate on all the sequence. And specificity of the restriction endonuclease is, and they can operate only in sticky end. Restriction enzyme only operate in the sticky end. And sticky end ಅಂದ್ರೇನು? Sticky end ಅಂದ್ರೆ ಇಲ್ಲಿ ಮಧ್ಯದ ಸಿರಿ, ಇಲ್ಲಿ in between, ಇಲ್ಲಿ ಮತ್ತು ಇಲ್ಲಿ ಹೀಗೆ ಈ ರೀತಿಯಲ್ಲಿ ಏನೋ ಬಂದರೆ, ಈ ರೀತಿಯಲ್ಲಿ ಕಟ್ ಆಗಿದ್ರೆ that is called restriction enzyme. So once again I am telling you, all the enzyme here generally what you call the restriction enzyme that used in cut the DNA molecule. Generally enzymes are two type. I said restriction enzymes are two type: restriction endonuclease and restriction exonuclease. And generally these enzymes are used to cut the DNA molecule. DNA molecule na cut ಮಾಡಲಿಕ್ಕೆ. Okay, and that is why here restriction enzymes or restriction enzymes generally called as a biological seizure, katri, or biological knife like chalk. Remember this word. In CET NEET may be asked. Maybe it is not given in your book, but you must remember that restriction endonuclease or restriction exonuclease, they are both what are called what here the biological life or biological seizure. ಕಟ್ ಮಾಡಲಿಕ್ಕೆ ಯೂಸ್ ಮಾಡ್ತಾರೆ. ಹಾಗೆ ರಿಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸ್ ಎಲ್ಲಿ ಕಟ್ ಆಗ್ತದೆ? ರಿಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸ್ ಓನ್ಲಿ ಕಟ್ ಇಟ್ ದ ಸ್ಟಿಕಿ ಎಂಡ್. ಎಲ್ಲೆಲ್ಲಿ ಸ್ಟಿಕಿ ಎಂಡ್ಸ್ ಇದಾವೆ ಹೀಗೆ, ಇಲ್ಲಿ ಸ್ಟಿಕಿ ಎಂಡ್ಸ್ ಇರ್ಬೋದು, ಹೀಗೆ ಸ್ಟಿಕಿ ಎಂಡ್ಸ್ ಇರ್ಬೋದು, ಅಥವಾ ಇಲ್ಲಿ ಕೂಡ ಇರ್ಬೋದು, ಇಲ್ಲಿ ಕೂಡ ಇರ್ಬೋದು, ಸ್ಟಿಕ್ ಹೀಗೆ ಸ್ಟಿಕ್ ಮಾಡಲಿಕ್ಕೆ ಆಗುವಂಥ ಎಂಡ್ ಇದಾವಲ್ಲಿ ಅದಕ್ಕೆ ಮಾತ್ರ ಕಟ್ ಆಗ್ತದೆ. ಬಟ್ ಎಕ್ಸೋನ್ಯೂಕ್ಲಿಯಸ್ ಇಸ್ ನಾಟ್ ಲೈಕ್ ದಟ್. ಎಕ್ಸೋನ್ಯೂಕ್ಲಿಯಸ್ ಯಾವಾಗಲೂ ಕಟ್ ಆಗೋದು ಬ್ಲಂಟ್ ಎಂಡ್, ಲಾಸ್ಟಿಗೆ ಹೀಗೆ ಇಲ್ಲಿ ಅಥವಾ ಇಲ್ಲಿ blunt end, ಲಾಸ್ಟಿಗೆ ಎಂಡ್ ಬರ್ತದಲ್ಲ, and that end is called a blunt end. And generally that restriction exonuclease has a no more important role in the biotechnology or genetic engineering. Genetic engineering ದಲ್ಲಿ the largely used enzyme is a REN, that is a restriction endonuclease. There are more than 900 different type of REN have been discovered so far from the different microorganisms. Got it? There are more than 900 different type of restriction enzymes have been discovered so far from the different type of microbe, then bacteria, etc. And so thereby what here the restriction enzyme has the vital role. This restriction enzyme has what? The vital role, and that is why it used in cut the sticky end. ಅದನ್ನು ಸ್ಟಿಕಿ ಎಂಡನ್ನು ಕಟ್ ಮಾಡಲಿಕ್ಕೆ ಯೂಸ್ ಮಾಡ್ತಾರೆ. ಹಾಗೆಯೇ ರಿಸ್ಟ್ರಿಕ್ಷನ್ ಎಕ್ಸೋನ್ಯೂಕ್ಲಿಯಸ್ ಅನ್ನು ದ ಬ್ಲಂಟ್ ಎಂಡ್ ಕಟ್ ಮಾಡಲಿಕ್ಕೆ ಯೂಸ್ ಮಾಡೋದು. ಅದಕ್ಕಾಗಿ ಬೋತ್ ವಾಟ್ ಎಂಜೈಮ್ಸ್ ಆರ್ ಯೂಸ್ ಟು ಕಟ್ ದ DNA ಅಥವಾ ಕಟ್ ದ ಜೀನ್, ಆ್ಯಂಡ್ ದಟ್ ಈಸ್ ವೈ ದೇ ಆರ್ ಕಾಲ್ಡ್ as a biological what? The knife or biological seizure. So thereby here enzymes play a very important role. Then the host. Generally your host is E. coli bacteria. E. coli bacteria. And why host is used? Because the host has an ability to replicate and divide, and this is a best the raw material for the production of the desired things. Production of desired things. So that is our host used. And one thing. Second thing is that every host like bacterium and that containing a plasmid. That contain what? The plasmid. So what is that plasmid? Plasmid is an extra chromosomal circular DNA. See, this is a bacteria and this is E. coli bacteria, and in this the common nuclear material is that is this is a nuclear material. This is nuclear material. Now, and beside this there is some circular DNA present, and that circular DNA is what? Plasmid. Circular DNA is what? The plasmid. And that circular DNA has a self-replicating power. That circular DNA has what? The self-replicating power, and because of this reason, here plasmid is used as a vector, self-replicating power 1, and that also has an ability to carry the foreign gene. Has an ability to carry the what? Foreign gene. Foreign gene ಅನ್ನು ಕೂಡ ಅದು ಕ್ಯಾರಿ ಮಾಡ್ಕೊಂಡು ಹೋಗ್ತದೆ. ಅದಕ್ಕಾಗಿ ಇದಕ್ಕೆ ಏನಂತ ಕರೆದ್ರೆ ವಾಟ್ ವೆಕ್ಟರ್ ಆಗಿ ಯೂಸ್ ಮಾಡ್ತೇವೆ. What is a plasmid? Plasmid is an extra chromosomal circular DNA, has a self-replicating power. The self-replicating power, and not only that, it has an ability to carry the foreign gene, and they also have the origin of replication property. So because of this reason, it is used as a vector. That is why plasmid vector ಆಗಿ ಕನ್ವರ್ಟ್ ಆಗೋದು ಯಾವಾಗ? So plasmid ಗೆ foreign gene has to be spliced, and foreign gene has to be blocked. Once the foreign gene is spliced ಆಗಿದ್ರೆ, and one more enzyme is used and herein, and that is called as DNA ligase. That enzyme is called DNA ligase, and that DNA ligase help in giving connection. ಅಂದರೆ ಫಾರಿನ್ ಜೀನ್ ಮತ್ತು ಒರಿಜಿನಲ್ ಜೀನನ್ನು ಕನೆಕ್ಟ್ ಮಾಡಲಿಕ್ಕೆ DNA ligase is used, and so that is why the DNA ligase is commonly called as a biological tailor. Okay, biological ಅಂತ ಕರೀತೇವೆ, tailor ಅಂತ ಕರೀತೇವೆ. And so these are about the enzyme, then host, and the plasmid you studied. And the next class you have to study the technique, bioreactor you have to study at the end. Next class we will study the real technique of the genetic engineering, and all that we will discuss in the next class.